Good afternoon. This is Marwan Abdellah from the Blueprint project at EPFL in Switzerland. Today I'm presenting our paper that is discussing new method for generating mesh models of astroglial meshes from synthetic morphologies for in silico simulations and visual analytics. I might assume some of the audience might know about the Blueprint project and its mission to build a brain in silico. It's a way to establish simulation neuroscience as a complementary research approach alongside the experimental, theoretical, and clinical neuroscience, trying to understand the brain by building biologically detailed digital reconstructions of its different components and then performing simulations down to molecular level. So what we are trying to do is to understand the function and structure and even the function from the structure. And to accomplish this mission, we need first of all models. These models are either extracted from optical microscopy stacks, the ones we acquire with the optical microscopes, or even the electron microscopy stacks that are available in the community. And then we run simulations, large-scale supercomputing-based simulations on these models, trying to understand what's going on. And then we use visualization at the end to provide an interpretation of what we can see, and also to verify the models and the results of the simulations on these models. This is, for example, full compartmental simulation of a spontaneous activity of a very thin slice that is reconstructed from the cortex of a 2-week-old rat. This is just an electrophysiological simulation that is entirely composed of neurons, but our brain is not just about neurons. The brain is consisting of other components, including blood vessels and also glial cells, and in this context, astrocytes. These three components form a functional unit, what's called the NGV ensemble. And this unit, it regulates the brain energy management, and it's quite essential for our brain to perform as it should be. The main focus will be given in this presentation to astrocytes. Astrocytes are similar to neurons. They do have arbors or branches and cell bodies or somata, but moreover they do have end feet. End feet are wrapped around blood vessels to provide all the energy metabolites needed to for the neurons to function correctly. Astrocytes are quite complex compared to neurons, although they are similar in structure, but they do have sponge-like structures that would make imaging them with the fluorescent microscope a challenging mission. And also their end feet have no standard way for storage, so it's quite difficult to find models for them. So astrocyte models are not as available as neurons, and unfortunately there are no tools for astrocytes to get them visualized or analyzed as well. In 2018, we presented NeuromorphoVis for analysis and visualization and even meshing of neuronal morphologies, and it has been a very successful tool that has been used widely in the community. And in 2020, we have extended the tool to work on vascular morphologies, very large-scale vascular morphologies for analysis and visualization as well. This year, we are focused on building mesh models for the astrocytes from synthetic astroglial cells for two purposes. The first one is in silico simulations, and the other one is mainly for visual analytics. The point is, astrocytes are not available in the neuroscience community as neurons, so we had to do some work on NGV modeling, with which we build a system or an architecture for the NGV system that would allow us to create and synthesize astrocytic morphologies that we can use later to build the astrocytic mesh models. These are 25 different astrocytic morphologies that have been generated by Eleftherios Desis. And if you want further details on the astrocytic generation model, you can refer to this thesis. So our pipeline that is based on Metaballs, it has the following workflow. So the astrocytes are being synthesized and the skeletons are generated, and our pipeline consumes these skeletons and uses Metaball scanning to generate a non-watertight mesh. This mesh can be used basically for visualization and visual analytics, but it cannot be used for simulation because it's non-watertight and it has quite few self-intersecting triangles. So we perform one step of optimization that reduces the polygonization, improves tessellation and topology of the mesh. And from this watertight mesh, we use any of the available tetrahedral mesh generators, such as Quartet or TETGen, to generate a tetrahedral mesh that can be used for performing reaction diffusion simulations. The morphology structure is quite similar to neurons. It's composed of two main components, samples and manifolds. So the arbors and the soma are represented by samples, where each two samples form a segment and a group of segments between two bifurcation points form a section, and a list of sections organized in a hierarchical manner form an arbor. Astrocytes have two different arbors, dendrites and axons, as shown on the right image. And also the manifolds represent here the end feed. So they are represented by triangular patches where every point along this patch has a certain thickness. Our approach is based on metaball scanning. So we create from the samples what's called the field. And from this feed, we create a metastructure that is being polygonized using one of the derivatives of the Martian cubes algorithm to create a mesh. So we use the same algorithm to generate the astrocytes, but we do some tricks to be compatible with the astrocytic morphologies. First of all, for the arbors, we cannot use the arbors that are generated directly from the synthesizer because the morphology has tens of thousands of samples, and that's going to take a lot of time to generate just one mesh. So we perform adaptive resampling, with which we don't lose any information, but we just get rid of the unnecessary samples. So we end up having hundreds of samples in one morphology. For the somata, we do have two methods to reconstruct the soma. We have a method that is based entirely on the metaballs, with which we use the mean radius of the soma that is reported in the loaded file, and we use as a guideline the initial samples of every connected arbor to it, and then we just grow the soma that way. This approach might be quite easy and naive, generate approximate shapes, but it's not the best. So we have actually presented another solution based on soft body dynamics and metaballs. It's sort of a hybrid approach from a technique we presented before in 2017. The point is we cannot use the objects that, or the mesh objects that have been generated from the soft body dynamics directly, because they do have non-uniform distribution along the surface of the meshes. To resolve this issue, we had to run another mesh generator that creates randomly distributed surfaces with which we don't have any gaps at the reconstructed mesh. As you can see in A, we don't have the vertices are not uniformly distributed, but in B, C, D, they are uniformly distributed with different tessellation levels. This gives us the capability of creating high quality or highly realistic somatic profiles and tweaking the soft body parameters such as stiffness and number of simulation steps to create as realistic as possible, or somata profile that could be realistic as possible. As a result, to compare the two methods, these are two meshes for the somata reconstructed from method 1 and method 2, and as you can see, the one that is generated with the hybrid approach is much more realistic than the one that is generated with metaballs only. The third component, which is the end feet, is provided, or like the end feet are provided as a manifold surface, a patch of triangles where each vertex has a corresponding thickness. Unfortunately, the tessellation of this patch is quite low, so we cannot use such patch as it is because it will end up having a fragmented mesh. Therefore, we have to subdivide the end feet to a certain threshold where no gaps can occur. Here we do have several subdivision levels, so we have to find a compromise between level needed for accurate tessellation and good performance. So we have selected the subdivision level of 2, with which we do have a smooth surface and we don't have any holes along the surface of the mesh. Okay, so putting all the components together, we end up having the following results. So from the Metaball scanning implementation, we have the mesh in red, where we when we polygonize the meta object of the astrocyte, we end up having this mesh that is highly tessellated, because the tessellation here is based on the resolution of the morphology or the smallest sample along the entire morphology, and it might have self-intersections. So we then use some optimization code that is written in C++ to adaptively optimize the mesh and reduce its tessellation, improves its topology, and ensures that the mesh is watertight. And in C, it's a physically based rendering in Blender using cycles, just to demonstrate how the final mesh would look like. Then we perform mesh analysis to generate the qualitative results of the mesh, of both meshes, to show the profiles, a comparison between the profiles of the meshes generated from the Metaball algorithm directly and the ones that are generated from the optimizer, to see that the optimized ones have very nice distributions for the dihedral angles and the radius ratio and radius to edge ratio, etc. We have used 5,000 morphologies, astrocytic morphologies, to test our pipeline, and the results were all successful on all the provided astrocytic morphologies. So we just here show the results for different astrocytic meshes. See how distributions would look like at the end. Okay, so in terms of applications, our results are directly applied in two domains. The first one is performing reaction diffusion simulations of the astrocytes to be able to understand the calcium dynamics of astrocytic meshes. So here, this is just a one-time frame showing the calcium signal along the surface of the astrocytes at different locations. And this is a time series showing the simulation results at different time steps. The interpretation of this simulation is extremely useful to let us understand the mechanisms of how the calcium is being propagated in the intracellular space of the astrocytic morphology. The other main application is visual analytics. So the meshes that are generated from our pipeline are combined with the vascular network in order to see the distribution of the astrocytes in the circuit and ensure that they are correctly placed in the circuit. And also we have to ensure that the astrocyte end feeds are correctly wrapped around the blood vessels, so they are not overlapping, intersecting, or even like falling far apart from the blood vessels. So this is important for us to validate the astrocytic morphologies that are generated within Cyder are correct, and also to generate multimedia content that we can use later for dissemination, as this rendering has been generated with cycles, so it's quite pleasant also that we can use it in technical reports or publications. Our pipeline is based on Blender, and it can be called from a simple command line interface, and it can be also configured with a batch configuration file. The code must be executed in a Blender context using its Python API, and this is the command line interface. Further instructions to run this pipeline are available in the supplementary material. The source code has been integrated into Morphologies, and it's entirely available on GitHub. So if you want to have a look on the code, you can just download it and let us know if you need any help to run it. In conclusion, we presented a new method for building astrocytic meshes from synthetic morphologies using Metaball scanning algorithm. And our implementation is integrated in a pipeline, an automated pipeline that is capable of creating watertight optimized meshes that can be used for simulating calcium dynamics based on one of the simulators, available simulators in the community like STEPS or Phoenix. Somata are reconstructed with two different approaches based on metaballs and a new hybrid approach that involves the reconstruction of physically plausible somata using soft body dynamics. Our results are applied in silico simulations and visual analytics as well, and the code is integrated into Morphologies, and it's open source on GitHub, so you can just download it. This work has been funded by the ETH domain, EPFL, and King Abdullah University of Science and Technology in KAUST in Saudi Arabia as well, under this award number. So thank you very much, and if you have any questions or you need any further details, please don't hesitate to contact me at marwan.abdellah@epfl.ch. Thank you very much.